Hello everyone and welcome to today's webinar hosted by Organovo and presented by Dr. Sharon Presnell, Chief Scientific Officer at Organovo. My name is Deb Nguyen and I'm the Senior Director of Research and Development Tissue Applications here at Organovo. Before we get started, I want to remind everyone in the audience that we will be holding a Q&A session following this presentation. We encourage you to submit any text questions at any time during the presentation by typing your questions into the questions pane of your control panel. We'll collect those questions and address as many as we can during the Q&A session. And with that, it's my pleasure to introduce Sharon Presnell, the Chief Scientific Officer at Organovo. Dr. Presnell holds a PhD in pathology from the Medical College of Virginia. And as the Chief Scientific Officer at Organovo, Dr. Presnell has led the growth and development of our R&D organization, including the evolution of the company's technology portfolio. Sharon?
Great. Thank you, Deb, for the introduction. So we'll jump in and get started here. So we're going to talk today about the use of Organovo's bioprinted 3D human tissues for the assessment of drug toxicity. Just reminding everyone, Organovo is a publicly traded company on the NYSE MKT tier. And so here's our release statement. So as we think about the drug development process, at every stage of the process we are using systems to answer questions that guide next steps in that process. And so whether it's discovery related, toxicity testing related, or efficacy testing related, we're using systems such as cell-free systems, cell lines that are grown in simple monolayer cultures, or animal studies to kind of predict or approximate that human response. What we're going to talk about today is kind of an addition to those more complex systems that take us away from animals but keep us in the 3D space, and 3D bioprinted human tissues help us to get there. And so we're going to talk today about how those systems help us answer some of these challenging questions in the development process in a human context. So if you think about the biological responses that we're measuring as we go through these processes, we're really asking about cellular responses, but we're trying to ask them in the context of the human being. And so you can imagine, if you look at these images here, the endothelial cell that's sitting on this polystyrene dish is probably going to give you a very different response to a biological modifier than, say, the endothelial cell that's lining the sinusoid in the liver. And certainly if you take that further to the organism where there's blood flow and other considerations and other cellular systems present and tissue systems present, you're going to get different responses. And I think this is a very obvious thing, but is really important to what we're trying to get at when we put cells together into 3 dimensions to better approximate the tissue environment. So what are the real key elements of tissue level context that we're trying to recreate when we put cells together and bioprint with them? So if we look at a native tissue, it's always composed of multiple cell types. There's not a tissue in our body that only has one cell type. And those cell types are arranged with each other in 3 dimensions, creating real biological mass, and they're compartmentalized based on the relative positioning of cells to each other and then the extracellular matrix that those cells produce in that context. And so when you look under the microscope, the patterns that are created by a particular tissue are ultimately dictated by spatial arrangement of the cells and the extracellular matrix that they have produced. And this is really hinges around the development of those intercellular relationships, and reestablishing those to the degree that is possible helps us better approximate that context of the tissue when we're trying to ask these biological response questions. So let's talk a little bit about 3D bioprinting and what that really means. So for us at Organovo, we're really focused on a few key elements of bioprinting. One is the fact that it's automated. So if you do make 24 little livers, there are going to be 24 little livers that pretty much look just alike and will function very much the same and structurally be the same. You're not relying solely on self-assembly to create your mass, and you have a little more control over where things are, and how reproducible they are from tissue to tissue. Because you can work with multiple cell types on the printer at a time, you can pattern those down relative to each other and have spatial control. So you can put, in the liver stellate cells where stellate cells go and hepatocytes where hepatocytes go, And you are able to control that as the user and create these geometric patterns. And that's really what defines bioprinting. It's the use of cells, it's spatial control, and it's the automation. So if we look a little more deeply at the process of bioprinting, kind of going from left to right here, so we're starting with cells, and we really focus on use of human cells, and we may work with primary cells, we may work with iPS-derived cells. We're pretty agnostic in terms of where the cells come from, but we're very, very focused on human systems and building human systems. Those cells are either isolated or isolated and expanded up and then formulated into what we call the bioink. And the bioink is predominantly for us composed of cells, and it may have one cell type, it may have 2 or more cell types in a single bioink, and there may be a hydrogel excipient included sometimes to ease the passage of those cells through the printer or to provide some structure. I'm going to show you here a little picture up at the top. So just to put this into context, one of the things that's very unique about the Organovo brand of bioprinting is that we're very focused on generating things that are 100% cellular at the time that they're used. And so if you look at a 35% cellular bioink or 100% cellular bioink, you see even at 35% cells, which is pretty high concentration by most standards, you're still not getting a lot of cell-cell contact in there. The cells are kind of embedded in a hydrogel, and this is shown pretty much immediately after printing, that difference. But the printer itself is pretty agnostic to that as well, and you can print anything from a 100% hydrogel all the way up to a 100% cellular bioink, and the system is compatible with that. The bioprinter itself is everything that touches the cells is made of biocompatible materials. It's multimodal. You can print by a variety of different mechanical means and generate structures of different sizes, shapes, and thicknesses with a high degree of spatial control. And what this gives you in the end, and I like to say, because I'm the biologist, this is not the end, it's the beginning, it gives you a 3D human tissue that you now have to figure out all the ways that you take care of it after it's made. And so we spend a lot of time on understanding the media requirements and all the aspects of how we maintain these tissues in vitro once they're made so that they maintain their function and that they give us a window in which to use them that's meaningful. And just to reiterate here, and you're looking here actually at a little plate of our liver tissues, at the time that we're using these, they're composed 100% of cells, and we're not working with fabrication methods that include the integration of scaffolds. So looking a little more closely at the ExVive 3D liver tissues specifically, so as we're going to talk about this particular tissue today, it's a fully human multicellular tissue. All the cell types are primary human cells. We work here with primary human hepatocytes, primary human stellate cells, and primary endothelial cells. And these cell types are patterned in specific zones within the tissue as it's printed, and this creates this compartmentalized architecture. Here you see a picture of the tissue grossly, just looking at it from above, and then if you look at the trichrome section below here, this is the non-parenchymal cell zone, which is clearly architecturally and compositionally different than the parenchymal zone, which is labeled with a P here. And so you can appreciate spatially where the different cell types are patterned down. One of the unique aspects of this, and we'll get into this a little bit in the next slide as well, is that these are durable over extended periods of time. And what this does is allow us to explore low chronic exposure models. And it's not just about the high acute doses of compound, but really asking those trickier questions. What happens if I expose this tissue to a toxicant or a potential toxicant for a week or a month in some of these longer time frames? So now just focusing on some of the attributes of the tissue. So, if you look at the upper left here, you can see with the green stain that outlines the cells, the junctions in between the hepatocytes, which are albumin positive, which is shown in red. And this just gives you a sense of the cellularity of the tissue. It's very tissue-like. Cells are touching other cells, and the hepatocytes have a very hepatocyte-like phenotype. If you look at the picture on the right-hand side here, this is one of my favorites. If you see the red staining there, those are the CD31 positive endothelial cells within the tissue. And the green staining are the desmin positive stellate cells within the tissue. And you see these nice quiescent morphologies in the stellate cells which develop within the tissue, and the approximation of those to the endothelial cells. And these intercellular relationships play a big role in both maintaining hepatocyte function and health, and allowing us to see things besides just acute hepatocellular toxicity. So we see all kinds of things in the tissue. We can measure things like cell loss and migration. We can look at proliferation. We can appreciate phenotypic changes, such as the activation of a stellate cell, for example. We can look at the production of ECM over time, or remodeling of that tissue over time in response to certain insults. We can look at cytokine or protein secretion, lipid accumulation, and glycogen accumulation. Both of these are shown in those lower panels there, and you can see the oil red O stain appreciating the fat accumulation in the liver, and also the glycogen here on the right. And we do get the question about the durability of the tissues from a functional perspective, and so if you look at ATP levels over time, we see that those are not only stable, but they actually rise over time after manufacturing. And here is just shown as a comparison the gold standard 2D cultures, and you see that those kind of wane over time, and that's a known fact in the literature. That's not a surprise, but we have them here as controls. You see something very similar with albumin, which is in the lower right here, and the protein production of the liver-specific protein is coming up over time and very stable in the system over time, and it's not a degenerating system. It's very dynamic, and it continues to function in a stable way over extended periods of time after it's fabricated. And this would be in contrast to a liver slice, say, that might look really good the first 24 to 48 hours, but then that's going to decline over time, much like the 2D cultures do. And then if you look at the upper right-hand side, this is cytochrome P453A4 RNA expression, and you're looking at a fold change here over time compared to day 3. And so what we're looking at is that the expression of this kind of difficult to maintain cytochrome P450 is actually stable and rising in the system as well. What's nice is if you look at the lower panel there, now we're looking at actual activity of that enzyme, so the ability of it to convert midazolam to hydroxymidazolam mediated through 3A4. And what we see is even at 28 days, the system is quite competent to create this metabolite over time. And so this gives us confidence that, yes, these tissues are viable, they maintain their morphology over time, and they also maintain some liver-specific function over time, and this makes us comfortable to test them in response to certain compounds. So when we think about modeling, toxicology phenotypes, again, as we alluded to a little bit earlier, the acute 24 hepatic toxicity questions are a little easier to ask. If you want to ask some of the more complicated things, what's the inflammatory response over time? Am I seeing necrosis or apoptosis? Can I see the accumulation of fat in response to this insult? Or ultimately, can I see something really complex like fibrosis? We're able to model these different phenotypes that are all relevant, especially in the chronic toxicological phenomenon. So we're going to step through some of these examples here, and if you look at this slide, what we're showing is that we can take, again, primary human Kupffer cells and integrate those into the tissues as well at the time the tissues are fabricated. And there's a couple of points to make with this. So here, the Kupffer cells have been incorporated, and so tissues with or without the Kupffer cells have been exposed to LPS, and we're looking at a very typical response to that with secretion of IL-1 beta on the left or TNF-alpha on the right. And there's two important things to note. One is that the tissues without the Kupffer cells present actually still have an LPS response. And the stellate cells that are incorporated in the tissue are competent for cytokine production and LPS response, and they really are perhaps a little bit of an underappreciated cell type in this regard. And in the base model that does not contain Kupffer cells, you can still see these type of responses, cytokine responses. But when you layer the Kupffer cells into the system, you can actually potentiate those cytokine responses and see a greater uptick in the secretion of IL-1 beta and TNF-alpha. And this gives us confidence that A, we can incorporate Kupffer cells into the system, and that's not a particular challenge, and that you can then really in a deep way examine the inflammatory response, and you could even think about doing subtractive experiments where what is the inflammatory response, what's the role of the Kupffer cell versus the role of the Kupffer cell with the stellate cell present, and vice versa. So we're going to look at a few case studies. Some of these were presented, like this one, at SOT and this past meeting of SOT in New Orleans. And so we're going to look first at acetaminophen, which is a well-characterized hepatotoxicant. It really causes oxidative stress and damage and depletes glutathione in the system and ultimately shuts down the production of ATP. And these things are kind of known about that drug, and it's pretty well characterized. So here, a 6-hour to 28-day exposure was conducted at various concentrations, and we looked at albumin, ATP levels, and glutathione levels. And so if we look at the next slide here, one of the things you notice in the graph on the left, so these are ATP levels over time, and of course as the ATP is falling off with higher concentrations, that's giving you a sense that the tissues are significantly damaged and shut down. With chronic exposure over longer periods of time, the sensitivity to that acetaminophen is coming down substantially, and so that's really demonstrated nicely in these curves where you see the depletion of ATP in a dose-dependent manner, and that sensitivity is going to come down as you increase the exposure time in the tissue, which is not surprising. If you look at the graph on the right, this was a neat aspect of the study. So here, there was a short exposure to acetaminophen of 6 hours, and then with the removal of that exposure to acetaminophen and recovery, you begin to see, first of all, you see the depletion of glutathione that you would expect to see upon exposure to acetaminophen, but with the removal of that insult, you actually are able to see the system recover after a 6-hour exposure. And interestingly, after a 24-hour exposure, you're not going to see that recovery, and you see that control there on the right-hand side. So we can see a very typical acetaminophen response in the tissue, and that's accompanied by the biochemical signals that you would expect to see associated with acetaminophen and the glutathione pathway. So next we're going to look a little bit at triglyceride. So this was the first FDA-approved PPAR gamma agonist for the treatment of diabetes. And there was a lot of preclinical testing done with this drug before it went onto the market, and none of those animal studies or human hepatocyte studies revealed the liver tox issue. When it did go onto the market, it was ultimately withdrawn after several cases of acute liver failure and replaced by safer drugs like pioglitazone. And the mechanism of toxicity here are really believed to involve some of these less prominent metabolic intermediates that may be specifically associated with the diabetic phenotype that the drug was being given to. So here we did a 28-day exposure at both a 5 and 15 x Cmax concentration and looked at several endpoints, including metabolite formation. So very importantly, so if we look by mass spec, we see with exposure of the tissues to the triglyceride, we see the parent compound, of course. We see the quinone metabolite formed by 3A4 and 2C8. We also see the glucuronidated form and the sulfated form of the drug show up, and that shows us that our phase 2 enzyme and sulfotransferase activities are present and accounted for, and that the metabolites that are suspect in causing toxicity with this drug are actually able to be formed in the system, and that's an important kind of check. And so if we look at the outcomes, in the graph on the right here, you can see that the triglyceride at both 5x and 15x Cmax significantly decreased the ATP levels in the tissue, and that was much less notable with the pioglitazone at the same equivalent concentrations. If you look at the histology on the lower panels there, so you see with the vehicle there's really not a lot of tunnel positivity, an occasional cell, which we see turnover in our tissue. There's a little bit of proliferation and a little bit of cell death in our tissues always. And we look at the pioglitazone, and again there's an occasional tunnel positive cell that you see. But when we look at the triglyceride, in particular in the area of the hepatocytes that are adjacent to the stromal cells that are printed, we see lots and lots of tunnel positive nuclei. And so this was a really consistent finding here and really gives us confirmation at the histologic level that what we're seeing are dying hepatocytes, which does show up as well in the loss of the ATP. So here we're going to look at valproic acid. And this is a mechanism of liver injury that is accompanied by steatosis traditionally. VPA is an organic acid that's used in the treatment of epilepsy, bipolar disorders, and migraine. About 10% of patients develop some ALT elevations during long-term exposure, and those are usually asymptomatic and resolved, but sometimes severe injury occurs, and you can see things like mitochondrial toxicity and a steatotic phenotype develop. Here we did a 14-day exposure over a pretty wide range of concentrations and looked at several endpoints, including histology and ammonia levels. And so if we go to the next slide here, you look at the ATP levels over time, and it really took pretty high concentrations of the drug to start seeing a shift in the ATP and ATP shutting down. If we look at the ammonia levels, this is really interesting because the liver, when it is compromised, will not be able to deaminate or detoxify the ammonia, and the ammonia levels rise in the tissues, and this can cause things clinically like hepatic coma. And so it's an important clinical biomarker as well. And so what we see in these tissues is that pathway shuts down, and it does so at a lower concentration than when you see the ATP levels really shut down, and that makes sense from a temporal standpoint. But we can actually see a dose-dependent rise in the ammonia levels in the system over time, largely due to the inability of the tissues to deaminate because of the damage. Now, if we look at the tissues histologically, we start to see some really interesting phenomena. And so if you look across the top panel here, you can actually see at the 200 micromolar, you start to see the appearance of some vacuoles by H&E, and these get larger and more prominent as you go up in concentration. If we stain these with paralipin, which confirms that these are lipid granules or lipid vacuoles and not just a generic phenomenon, we can actually see the brown staining here, confirming that these are lipid granules in the tissues. And if we look at a trichrome stain of these same sections, what we see here is a little bit of a fibrotic response maybe starting to begin at the 200 micromolar concentration, and you see the granules present, the lipid granules present. And if you look at the 1 millimolar concentration, you can actually see some of these very big ballooned hepatocytes, the presence of some megamitochondria, and really a phenotype associated with kind of oxidative stress and damage in the system. So this is very encouraging. Again, you get the biochemical results, and you also have enough tissue mass that you can do this type of histologic assessment and see these other phenomena that would be difficult to appreciate if you couldn't look at the tissue along with the biochemical data. So lastly, we're going to go through an example of drug-induced liver injury with fibrosis, and this is some really interesting work that we did with the Hamner Institute for Health Sciences in Research Triangle Park, and with a really talented graduate student there, Leah Nerona. And so what we were really focused on here is in this fully human system where we've got not only hepatocytes and endothelial cells, but we've got stellate cells that in the tissue acquire a quiescent-like phenotype, which means that if you do something to them to activate them, you'll actually be able to see that. In a traditional two-dimensional culture, the stellate cells are culture activated, and it becomes very difficult to see them activate further with any kind of an insult or damage. And so having them incorporated into the tissue gives us this ability now to ask the question, does this insult include hepatic stellate cell activation, and could that then progressively go on to develop fibrosis? So here we've done a 7 to 14 day exposure at a couple of concentrations of TGF-beta, and TGF-beta is a known prototypical fibrogenic modulator, and this was included just to make sure that if we put something in that should absolutely be fibrotic, that we could see that. And then we also tested 2 concentrations of methotrexate shown here, 0.1 micromolar and 1 micromolar, and examined a variety of endpoints. So biochemically, if we look at LDH release, which is a signal of basically tissue damage, leakage of that, if you look at the purple dots along the bottom, this is the TGF-beta, and over the course of treatment, the TGF-beta did not cause really problems with respect to LDH release. There was not a sign of toxicity. This is in contrast to the blue dots that you see here, the concentrations of methotrexate. And at the later time points, we do really see an increase in LDH release, signifying that the tissue is becoming damaged over these longer periods of exposure to the drug. If we look at a snapshot at day 7 with the tissues, Leah was able to see some upregulation of collagen and also smooth muscle actin. And this gives you a hint that, well, maybe there's something going on there with the methotrexate and with the TGF-beta, and that led us to take a look histologically. So now if we look at these tissues histologically, at day 7 you begin to see some dissociation of the tissue with methotrexate, and this is a typical kind of early sign of damage in the printed tissues. And maybe there's a little more collagen starting to develop around the edges here. And if you look at day 14, you have a very prominent fibrotic phenotype. There's very large areas of nodular fibrosis. You can see bridging fibrosis between a couple of different nodules of fibrosis, and if you look closely amongst the hepatocytes, you see a little better on the bottom. You can actually see paracellular fibrosis developing between the cells, and if you look at the hepatocytes that are immediately adjacent to the nodular fibrotic areas, you can see some phenotypic changes very consistent with the clinical sample that you see on the left here. So this was a patient that was actually biopsied because of fibrotic change in response to methotrexate. And so you can see some nice parallels, the nodular fibrosis, the glycogenated hepatocyte phenotype, and many of the same phenomenon in that tissue. And so just to summarize what we've looked at here, you can really, in using the bioprinted tissues, appreciate a very diverse array of toxicological phenotypes within the tissue. You get the biochemical data, you can look at gene expression, there's enough biomass in these tissues, you can isolate microsomes, you can isolate enough RNA to do an array, and you can look at a lot of things, but histologically you can appreciate a lot of phenotypes here as well. So with LPS, we looked at inflammation. With acetaminophen, we saw necrosis and acute hepatocellular injury. With tribalism, we saw apoptosis. With valproic acid, we saw a steatotic phenotype, and then with the methotrexate, a fibrotic phenotype. So what this leads us to, when we're developing any of the tissues that we work on, we're really focused initially on getting the biology right, developing a liver, a good liver. We can use that in preclinical safety, look at things like hepatocellular toxicity, focusing on the chronic low-dose exposures, focusing on things that are mediated by indirect mechanisms, so things that may require a Kupffer cell or a stellate cell to mediate their injury. We're able to see those types of things and able to see injury that's atypical at the histologic level, so being able to really appreciate progressive steatosis or fibrosis. Once you have that disease state, now you can think about thinking of these tissues in efficacy and disease modeling and applying them as a test bed for drug discovery, biomarker discovery, and ultimately as we learn how to make larger and larger tissues, we can think about transplanting tissues as well. So with that, I think we're ready for some Q&A.
Yes, thank you so much, Sharon. This is really a great summary of our system. So now we're going to begin the Q&A segment of the webinar, and as a reminder, you can still submit your questions through the questions pane in your attendee control panel. So let's get started. There have been some great questions coming through from the audience. So I'm going to start off with a basic question about the size and shape of the tissue overall and its dimensions. Can you speak a little bit to that?
Sure. So our standard liver tissues are about 2.5 millimeters by 2.5 millimeters in X and the Y direction, and about 0.5 millimeter thick in the Z direction.
Okay, great. Another question that was followed up on that is, does the structure have finer structures like acinar structures within the tissue?
So it's not going to have the exact same I would say histologic pattern as an X-sized piece of liver tissue because, obviously this is an approximation. There's not going to be sinusoidal blood flow in these tissues. But what we do have is a compartmentalization within the tissues. And so there will be fields of hepatocytes that kind of approximate the lobe and then areas of non-parenchymal cells that kind of approximate more the portal zones.
Great. In a follow-up to that, there was another question that went around that same vein towards things like big biliary transport or canalicular networks. Can you speak about those in our tissues?
Yeah. So we do see some focal expression of some of the canalicular transporters in the tissues. We do not incorporate cholangiocytes in the tissue, and so they're not going to be bile ducts in the tissue today or anything like that. I think that answers the question.
Yeah, absolutely.
We do see polarization of the hepatocytes, and I think that's the fundamental question.
Yeah, absolutely. And to follow on from that, you mentioned the non-incorporation of the cholangiocytes. So there was another question from the audience here about other immune cell types. We spoke to the fact that the tissues can be stimulated to produce cytokine following LPS, but that's only one portion of the overall, immune function of the liver, for example. So what about some of the other immune functions that might be in native liver?
So we haven't, other than the Kupffer cells and the stellate cells, we really haven't played around with incorporation of any other more systemic, sourced cells that could end up in the liver through different mechanisms, more related to blood flow and the presence of that in the system. That's something that we would be very interested in going down the road, but today the focus has really been on stellates and Kupffer cells.
Do you think that the technology has any limitations there? I mean, you spoke to kind of the agnostic nature of the bioprinter. Do you think that we have any limitations that you're aware of today as to trying to incorporate those cell types there?
Oh, no, not at all. No, not at all. It's just a matter of finding the right application, the right driver for doing that, and then executing on it.
Yeah.
So partners are welcome.
Yeah. That's great. We talked about the size of the tissue. You mentioned that you're looking at, in millimeter sizes, kind of thickness in the X and Y, and even many hundreds of microns in the Z. So given that size, there have been questions about necrotic cores in our tissues, about problems with nutrient diffusion. Can you speak a little bit about our experience in that area?
I think with all the tissues that we make, speaking more broadly than just the liver, I think we spend a lot of time optimizing the bioink formulations and the geometry to ensure that that is not a problem. Certainly in the self-assembly world, when you just allow cells to come together as aggregates, you can hit a limit of where that stops working, right? And I think one of the benefits of bioprinting is that you can go in and create compartments that have distinct compositions, and those can help anchor a tissue. They can provide nutrients within the tissue. There's a lot of what I call paracrine or juxtacrine, stimulation that happens in our tissues, and the incorporation of cells like endothelial cells are really key to us not having those type of phenomena be a problem.
Yeah. We also talked about, you talked about the fact that at our time of use we're talking about being scaffold free and not having permanent scaffolding present in the tissue. Can you speak a little bit to the importance of that, why that's kind of a fundamental part of our approach?
Well, if you, our goal is to approximate native tissue. And for tissues like the liver especially, if you go in, take a biopsy of a liver and look at it, it's mostly cells touching other cells and a little bit of extracellular matrix that they make. It's not like it's 70% extracellular matrix, unless it's a very sick liver, and a few cells sprinkled within, right? So we're trying to recreate that native gestalt. And so that means mostly cells touching other cells and the matrix they produce. And for hepatocytes in particular, if you want to start an hepatocyte down the path of dying, detach it from other cell types, and that happens very quickly. And that's a very well described phenomenon. So those cell-cell contacts are critical not only to survival, but to proper function, polarization. There's a lot of growth factors that a non-parenchymal cell makes that the hepatocyte absolutely depends on.
Great. And what are some of the ways that we measure that interaction? So we're talking a lot about making sure that the cells have the ability to kind of interact with each other on kind of a technical level. What are some of the ways that we go about measuring whether that interaction is taking place?
A lot of it is functional. So, when we're first starting to try to print with a tissue or formulate a bioink, the failures, as you know, are kind of in your face, right? It comes together and forms a tissue or it doesn't. And so some of the outputs are very simple, like just adhesion and kind of cohesion and forming of a tissue, and that's a pretty simple thing to ask. At the end of the day, though, it really becomes about function, and I think that's where we spend the bulk of our time, is if you have the mix of cells right, you can get something that looks, feels, and functions very much like a liver, for example, and if you get the mix of things wrong, it doesn't turn out that way.
Yeah.
You get something that's more pathologic. If you put, if you stray too far from the native tissue ratio of cells, you can get some things that don't work out.
Yeah, I always think back to some of the histology that we show, like especially that ECadherin, right, where we're looking at the way that those cells are forming junctions with each other to tell us that they're kind of coming together and forming the right connections that we would hope that they would be showing.
Oh yeah, and that's a good point that you were probably making before. So it's the histologic assessment. Our ability to look at things histologically and say, is this looking like native tissue or is it not looking like native tissue? And those intercellular relationships are hard to discern. I would say not able to be discerned in like a traditional 2D system. You really have to have the 3D architecture to be able to do that.
Yeah. There are two other questions that came up in the list that I think are a little bit related, just on opposite ends of kind of the culture spectrum. So one was about, in this process of coming together, how long does it take the tissues to really be ready to start interrogating them following the fabrication? And then how stable is that structural integrity over a span of time?
So that's going to be a little different. The time it takes for something to be ready is going to be a little different depending on what tissue type it is. Taking the liver as an example, from the time the printer finishes it until the time that we feel like it is A-OK to put drug on it is about 60 hours. There may be some applications where you want to do that at a different time frame. The system is very dynamic. And so in that first week or so, you're going to see lots of formation of the microvascular structures, and you're going to see the stellate cells go more and more quiescent because they're kind of coming into themselves there, right? So depending on what the application is, you may choose a different time point. And I think that's very customer specific, right, what their needs are.
What about the stability over time? You mentioned this great work dynamic. So can you talk a little bit about what you mean by that in our tissue context?
I don't look at our system as being static in that, it's not like a liver slice where you take it out and there are the cells in the configuration that they're going to be until that liver slice is done. Ours is some strange blend of kind of cell biology and developmental biology, but in an adult. And so you've taken these cells, which are adult cells, and put them together, and they're forming their own milieu. And so that's going to happen over that first 60 hours, and it's going to continue to evolve for some period of time. And just like at any given time in our body, if we were to biopsy our liver right now, we would see some dying cells, some proliferating cells, some cell migration, and all of those things are going on in our printed tissues as well at any given time. And functionally, I think that's just how living tissues are, right? So once we see the albumin production and the P450 activities kind of come to where they're going to be after that 60-hour time point, those are stable over the course of the use of the tissue, which is typically for us around a month.
Great. I want to go back and touch on some of the cellular inputs based on some of the questions coming out about the choice of you focusing on human cells and building human-specific systems when a lot of the preclinical studies that are out there are done in rodents, for example, or other preclinical species. Can you speak to that a little bit?
Yeah, that's a great question. I think the reliance on rodent systems has been a necessity, but it's also been a challenge because those systems from a species specificity have not provided the evidence that triglyceride is going to be a problem in human beings. And so, we're still a small company and focusing on our first product, and where can we make the most impact? That's the question. And human translation is the challenge. So for us, making a human three-dimensional tissue system that can answer that question of, does this recapitulate human biology? That was the primary driver. No. It's not to say that the animal systems aren't interesting or there wouldn't be compelling experiments to do there, but the human system is absolutely requisite even in order to do those and to make a conclusion. Yeah.
Are the human cells that we use usually from the same donor within one tissue, or are they from different donors?
That's a great question. So today, those cells are going to come from different donors because of the way that the infrastructure is around getting access to human cells, right? So your average cell vendor is going to maybe be able to get you hooked up with hepatocytes, but they're not necessarily going to be able to also provide you with endothelial cells and Kupffer cells and stellate cells, and that's something that as organizationally we are working on changing. But today that's a necessity to use different donors.
Do you see any downsides to doing that, or do we have any concerns about using them from different donors within one tissue?
I think not for the applications that we're pursuing today. To me, that's not a big concern. Are there opportunities or applications where it would be compelling maybe to have cells all come from a specific donor? I think especially as you think really downstream and you're thinking about implantable tissues and things like that, then absolutely. And there may be some immunomodulatory questions where you would maybe want those things to come from the same donor.
Mm-hmm.
And so there are lots of compelling reasons to think that would be a good idea as we go forward.
That makes sense. So one other question that was coming in is talking about our system, and our audience is fairly savvy, and they definitely buy into the fact that having multicellularity and three-dimensionality is probably a better native system. Can you speak a little bit about the next generation models really quickly, looking at some of the other spheroid technologies or organ-on-a-chip technologies, and where you think we kind of all fit in together into this question of better modeling?
Yeah, I think there's applications for all those things, right? In different levels of throughput, it depends on what you're trying to ask. You know, when I think about the real differentiators of the bioprinted tissues, I think about our ability to compartmentalize tissues spatially. And because of that, then, the amount of biomass that we have and the ability to ask really complicated questions like progressive fibrosis, progressive steatosis, in the context of the spatial relationship between the stellate cell, the endothelial cell, and the hepatocyte. And that's really, to me, where the bioprinted tissues offer an extraordinary value, is in the chronic progressive injury scenario.
Great. Well, that's unfortunately all the time we have for our webinar today. There's been some great questions. Want to make sure we thank Dr. Presnell for all of the education today. Please look for a follow-up email with a link to view the recording of our webinar once available. Also, I'd like to mention to the audience that we've extended our 50% promotional offer on the ExVive liver model services through the end of July, which is great. So please contact us or visit the Organovo website for more details. On behalf of Dr. Presnell as well as the rest of the team at Organovo, thank you so much for joining us today, and have a great rest of your day.